Hello. In this lecture, we are going to we're going to derive a mathematical model that integrates some of the basic concepts of immunology. And in doing so, we're going to have our first mathematical representation uh, on the interaction between target cells, infected cells, and virions or particles of virus. These equations are going to look very familiar to you since they should remind you of some of the equations, some of the mathematical models that we had studied for species that were interacting with each other, and even some of the equations for evolutionary game theory. Um, they could also give you flashbacks with the equations of epidemic models. So this was one of the last graphics we discussed when we were talking about the clinical profile of HIV. And there were important quantities that we referred to. One of them is these these cells called the CD4 cells. And for a patient that is HIV positive, these are typical measurements. So remember, look at this graphic as a function of time on the horizontal axis, and what we're seeing here in two different colors are measurements on the viral load or the amount of virus at each time point, right? So you want to think of what's happening here. That would be a measurement that has to do with a time, I'm going to call here T sub 1, right? And then at T sub 1, there will be another measurement for CD4 cells. So every patient, a given patient, these are things that clinicians, lab technicians would do, or physicians, they would take a blood sample and they have a way of reading or measuring from this blood sample and coming up with a number with how many of these cells the patient has and how much virus the patient has for a particular time point. Okay, so this is the motivation to then write equations to describe the population of CD4s and the population of virus in a typical host inside one patient. So before what we're going to write here are some equations that will this is the most basic model, so this model doesn't have an immune response yet, but we'll come back to the CD4s after we derive this model. However, here we have some important basic ingredients. Okay, and by ingredients of the model, I mean that first we have a cell that is to be invaded. Okay, and so here we denote the virion or the piece of virus as a dot. And this addition sign or this summation is in the spirit of chemical reactions. You may think of this uninfected cell reacting with this particle of virus, and once the virus, remember that thing of the life cycle of a virus, how the virus finds its way and attaches itself to the surface of the cell and eventually breaks through the membrane and find its way to the nucleus and so on. So this representation is also depicting here that scenario in which now the cell has been invaded by virus, and this is going to be considered infected cell. So we have three things going on here. This empty cell is what we call uninfected or a healthy cell. This cell has not been infected, is uninfected. Then we have a population of virions of particles of virus that are interacting, actively looking for these empty or uninfected cells because they, remember, the virus is a parasite and they want to use that cell as a host. Okay, and then we have the population of cells that are invaded, and we call them infected cells. So now we're going to write some equations. We're going to say, okay, we have three populations to keep track of, and we want to think of the size of each population as a function of time. And specifically, we're going to write an expression for the rate of change on the size of uninfected cells, right? So this here is for cells that are uninfected. And the rate of change on the size of the population of uninfected cells is given by a constant rate of growth. There is this rate d, which this is a very standard assumption. This typically, in the absence of this term, we would say that d is positive, and therefore this is like a natural decay rate for those cells, or constant that denotes the mortality when the cell attains its lifespan. Okay, so it's the constant birth and the death of these cells are proportional to the size of that population, but that happens also at a constant rate. Then the next term um, has to do with encounters between X and V, where V denotes, so here we're going to have three state variables. The uninfected cells are going to be denoted by X, but really we mean that X is a function of time. Okay, and then we have virions, and those are going to be denoted by V of T. Okay, and then we're going to have the infected cells, which are represented here, and so the state variable representing them is denoted as y of t. So we have x of t, v of t, and y of t. So now let's look at the equation that represents the rate of change of the size of infected cells, and we can see how the interactions between uninfected cells and virus are detrimental to the population of uninfected cells. However, those interactions are precisely what makes the population of infected cells grow. Okay, and then we have a term that denotes the decay, natural decay, for those infected cells. Okay, and then what we have next is an expression for the rate of change on the population size of virus. It is stimulated, the virus grows proportional to the amount of infected cells, and then there is a constant U here that denotes the decay, some type of natural decay for the population of virus. Okay, so dx, ay, and uv are terms that mathematically speaking are analogous because they denote some type of exponential decay. Now, in the context of mathematical models, it is important to keep in mind the definition of reproductive number. In this case, we're interested on the ability of reproduction uh, for the pathogen. So we mention, or we say something like the R0 of virus, or basic reproductive number of virus, which means in this context the the expected number or average number of newly infected cells that are yielded by a single infected cell at the beginning of the infection. And it is assumed that at the beginning of the infection, most cells on that host are uninfected. So for the mathematical model that we just derived for those three nonlinear differential equations, R0 is an expression that involves lambda, beta, and k in the numerator, divided by the product d, a, and u. And as you may remember, this rates, D, A, and U, are the rates of decay for each of those populations. Well, we can discuss in more detail methods for calculating R0, but uh, let's let's um, let's put that on. Let's keep that as a side note, and let's note the following: the reason why this quantity is it's important is because it allows to have some type of threshold behavior for the dynamics of this model. Okay, so we have one behavior when R naught is exceeds 1. In such case, the in such case, we would say that on average, one infected cell gives rise to more than one infected cell, and therefore we would have an expansion of the infection. We would say that the infection um, spreads. On the other hand, okay, and this is a graphical representation. So we are going to pause and we will continue on the following part of this lecture.